Okay, selamat petang, good evening. Welcome to tuitiontoyou.com. You are with Will Sundram, and today we are having a special class to discuss what is the possible question for this year paper 3 biology paper. Based on the information that is circulating in the social media, I have created a sample question. What I feel could be the question this year. So if you already have the question, good. If you don't have, after this, I'll show you the link for you to download the question. Okay, so at the moment, you are done with your physics, you're done with your chemistry, and from the message that I received from your teacher, board question is easy. So now the bio paper is the decider. Now it's going to be all three easy question, or is the bio going to be the killer paper 3 question? We really don't know. At the moment, the information that we have, more or less, we know what is the experiment. No one knows the question. We are predicting the question. So maybe it's like this. And based on the information, based on my experience, I have created a sample. I normally, I always like to create my own question. So I have created a sample question for you, which I already forward into the group. If you don't have the question, you can later download from my website. I'll show you the link after this. So that is not the question. No one knows the question. What I feel could be the question, I have already created a question set. So hopefully you already download and use it as we are discussing the paper. Okay, so let's start. I think this class will be maybe less than 1 hour. Quite a compact explanation. So I think within 1 hour should be able to finish. Less than 1 hour should be able to finish this class. Okay, let's start. I hope you have the question paper with you. Okay. Okay, so this is the question paper. Minute. And most of the students are still in the tuition center, I think. Someone said, do it tomorrow. So tomorrow public holiday. I thought, no mind, finish it early so that, you have time to do your revision. Okay, so this is the question. In case you don't have the question paper with you, I think most of you have because I already put it in the group. But in case later when I'm showing this in the YouTube, if any student don't have the question, what you can do is you can just scan this, whether the question is bilingual. You can scan this QR code. It will lead you to a course in my website and register free registration, and then you can download. Once you register for the course, you will see this list there. And in that course, actually, you're supposed to, the one I'm going to discuss now is basically, the one I'm going to discuss now is this, Form 5, Chapter 2, Photosynthesis, light intensity affecting the rate of photosynthesis. In that course, you will also all the other paper 3 questions that I've discussed every year. Before the actual SPM question, before the actual paper 3 question, we'll discuss like osmometer is for your trial, calorimeter is last year paper 3, yeast fermentation was 2 years ago paper 3. So all the questions and the videos that I discussed the question is all there in the course. If you want later, you can register. At the moment, you already have the question paper. After I complete this video, I will also have the video uploaded into this course. So you can log in here to watch the video, or you can also go to YouTube. But at the moment, YouTube I haven't uploaded. Later I'll upload. So this is the QR code for the English and BM. Whichever, if you feel necessary, if you want to check out all the other paper 3 questions, feel free to register and then download the questions and watch the video. We are now discussing getting some messages. Okay, so this is the question we are going to discuss. It's 45 minutes. So we are expecting photosynthesis question. So by right, if you have the question set with you, you can go through the first page. So I have a list of apparatus and material. You can go through all of this. Since it's related to when you start the experiment, go through the list. I'm sure by now you already have the experience of doing paper 3. You are done with physics, with chemistry. Go through the list of material apparatus, especially if you have a mechanical device like a stopwatch, for example. Check whether it's functioning or not. Go through one by one. I think they give you around 5 minutes to check. So check each one of it. If you feel any of the mechanical device is not working properly, maybe you can ask them to replace. So as we go through this list of material and apparatus, we should have a general idea what are the variables. When it comes to paper 3, it's all about variable, manipulated variable, responding variable, constant variable, and most of it you can already identify based on the list that you have, material and apparatus. So at the moment, there are still students coming in. Okay, so at the moment, some of the more important ones that you need to take note is 0.2 sodium hydrogen carbonate solution. This is the one that is going to provide carbon dioxide for the plant to do photosynthesis. The plant itself is important. You don't have the plant, you can't do the experiment. I'm just going to highlight the main ones. What are the main ones? The ones that are related to the variable, constant variable, manipulated variable, and also responding variable. Hydrilla is constant variable. The type of plant is constant. The concentration of sodium hydrogen carbonate solution, this is also considered constant. Scissor is to cut the plant, retort stand. Okay, the light bulb is important. This is another constant. In every experiment, you must use, when you repeat the experiment, you must use the same voltage, which is 60 watt. Stopwatch is important because this is another constant. We don't know what is the experiment, but generally you're going to count the number of bubbles every 2 minutes. So the 2 minutes is supposed to be constant. To measure the 2 minutes, you need stopwatch. You need to fix the distance between the light and the plant. That is also constant. And the others here, thermometer, if you need to measure the temperature of water, if you need to measure. Actually, in my experiment, I didn't mention anything about temperature. If the experiment you're going to do, if you need to measure the temperature of the water bath, then you can use the thermometer. And another important one, this is something new, supposed to be dry cell, not dead cell, dry cells. Batteries 1.5 volt. This is what's new. So this is the mystery ingredient in the experiment that we have. We really don't know whether it's going to be there or not, but news circulating in social media, the experiment will have dry cell. So that is something new. Normally, when you do this photosynthesis experiment in school, you don't use dry cell. You just light up the light by fixing it to a power point. That's it. Power socket, you just switch it on, you get the light. But here, they are giving batteries. So the batteries now is the mystery. Why do we need battery in a bio experiment? If it's physics, understandable. How come bio got dry cells? So based on the presence of the dry cells, I've created an experiment, and also based on one of the state paper, I think they had something to do with dry cell. So using this information, I've created a sample question for you. Don't know whether that is the question. I feel the question should be like this. But always, better safe than sorry. Don't just fully depend on what I'm explaining. Also, read up your textbook on how to do the other experiments related to photosynthesis. So, and actually, the main idea of this video, this class is to show you the technique of answering, what you need to have in the answer to make sure you get the marks. End of the day is variable. Every single answer, you must have the variable. Let's start to do the question. So this is the question. Green plant need water, light, carbon dioxide to carry out photosynthesis. And based on diagram 1, plan an experiment to study the effect of light intensity on the rate of photosynthesis. When you see the word to study, to determine, to experiment, this is important. So you preferably underline this. To study the effect of light intensity on the rate of photosynthesis. Immediately from this sentence, we can already identify that the MV is manipulated variable is light intensity. immediately you can identify. Manipulated variable is the light intensity. This small. So this is manipulated variable. Responding variable should be the rate of photosynthesis. So responding variable is the rate of photosynthesis. The question itself already give you a general idea what's the experiment about. Is it light intensity? Is it concentration of carbon dioxide? Is it the temperature? From the first question itself, you already know this is what the experiment is all about. As I told you earlier, we don't know what's the experiment. I feel it is the light intensity. So I've created a question based on this. Later, after you already understand how to conduct this experiment, always read your textbook for the other two experiments related to photosynthesis. So at the moment, I'm focusing on the light intensity. This is how the apparatus setup is. One thing that I want you to take note is, this is the hydrilla plant. This is the hydrilla plant. Make sure the part that you cut, this is where you cut. When you cut, also preferably cut it slanted. Don't cut it across. Cut it slanted so that there is more surface. Just add in the student. Okay. So when you cut, make sure you cut it slanted so that there is more surface for the bubbles to be released. And make sure the part that you cut is facing upwards. If you face it downward, you're going to get very less gas bubbles. So make sure it's facing upwards. That means when you apply the clip, make sure you put the clip at the top of the hydrilla. So that the clip is to create weight for them to sink. So you want the top part to sink, not the part where you cut. So put it upside down. The clip must be at the bottom to pull the hydrilla to the bottom. So this is important because if you place it downwards, then you're going to get less air bubble. The experiment won't be that precise. One assumption, I'm just informing you the assumption. By right, they don't ask in the exam, but I want you to know one of the assumptions that we are making here, two assumptions that we are making here is the gas that is being released is oxygen. The gas that is being released is oxygen, purely oxygen. That is one assumption we are making. It might be oxygen, it might be carbon dioxide also. Because the plant is also doing respiration. Photosynthesis release oxygen. Respiration release carbon dioxide. So one assumption that we are making when we do this experiment is the gas that is being released is only oxygen. In truth, in real life, is not true because it might have oxygen and carbon dioxide. That is one assumption we are making, that the bubble is purely oxygen. Because it's purely oxygen, we can use it to determine the rate of photosynthesis. But the actual truth is, it's not purely oxygen. It might have carbon dioxide also. As I'm going through the explanation, if you have any question, feel free to use the chat panel to ask question. Or just switch on your microphone and ask question. Anytime you can stop me. If you have any question, any doubt, ask. Clear it before the exam. So one assumption, we assume the bubble is purely oxygen. The second assumption is all the oxygen that is produced through photosynthesis is released by the plant. That's the second assumption we are making. Semua oksigen yang dihasilkan melalui fotosintesis dibebaskan keluar, which is also not true. Why not true? Because some of the oxygen is used for respiration. Some of the oxygen is used for respiration. It's a living plant. They are doing respiration, meaning they use the oxygen. So by right, they produce a certain amount of oxygen. Some of it is used for respiration. So the number of bubbles released is by right lower than the actual amount of oxygen produced. I'm going a bit fast actually. If I'm going too fast, just put a note in the message. So you're going too fast, slow down. Sometimes I get excited, I tend to, rush a bit. So the two assumptions, I repeat, the first assumption is we assume the air bubble has only oxygen, which is not true. It might have oxygen and carbon dioxide. The second assumption that we made when we do this experiment is all the oxygen produced by the plant through photosynthesis is released. That is also not true because some of the oxygen is used for respiration process. In case, someone, I was talking to one of the student earlier, he said, sir, chemistry had K-BAT question. I asked him, what do you mean by K-BAT? They asked something, he said something like prediction or something like that. So what if, suddenly this question out of nowhere, they ask you, what is the assumption you made? Who knows? You're supposed to know the assumption. So if they want, they can ask. So I'm just informing you in case they ask, this is the two assumptions. There are many more, but these are the main two when we are doing this experiment. Okay. So you can take this as the precautionary measure. Facing the cut stem upwards is one of the precautionary measure. Okay. So by right, I think for bio is going to be guided. Because I think one of the physics or chemistry already unguided, meaning most probably bio will be guided. I don't like to, in quote, gamble. Whether guided, unguided, I don't know. I'm going to explain to you the procedure, the full procedure. If they already give you the procedure, fine. If they didn't give you, then I'm going to explain to you the procedure. By right, you should know how to do it. So let's look at the question. This is the same code that I showed you earlier. So to study the effect of light intensity on the rate of photosynthesis. Okay, now we go to the first question. Plan your experiment by using the apparatus and material provided. Your procedure should include method to handle the variable and precautionary measure. The precaution is this. We'll have it in the procedure also. The precaution should be in the procedure. As I'm explaining the procedure, I'll highlight to you what is the precautionary measure. We'll have two precautionary measure. I'll highlight in the list. Okay. So here is 4 marks. Basically, end of the day, the examiner is interested with the variable. When you are conducting the experiment, you must inform the examiner how do you control manipulated variable, how do you get the responding variable, how do you make the constant variable constant, and what is the precautionary measure. They already inform you, right? And straight on your face, this is what we need. So they are specifically interested in the explanation of the 3 variable and also the precautionary measure. We know from the heading earlier, you are going to study the effect of light intensity. In your procedure, you must have an explanation. How do you get different light intensity? The experiment you have in the textbook, basically what you do is you change the distance. You change the distance between the light bulb and the plant. 20 centimeter, 30 centimeter, 40 centimeter, 50 centimeter, 60 centimeter. The logic is the further the light, the lower the light intensity. That is what normally we do in class based on the textbook. But because some rumors that there is going to be battery, I have modified the experiment. We are not changing the light bulb. We are going to fix the position. The light bulb, we are going to use different power. So we are going to give maybe 2 batteries, maybe 4 battery. The more battery you have, the more power the bulb gets, the higher the light intensity should be. So it's just a modified version of the light intensity. I remind again, I don't know what is the question. No one knows the question. We are just predicting. Maybe it's like this. So instead of changing the distance, you are going to change the number of light bulb. So in your procedure, you must have that explanation. How do you change the light intensity? So use 2 batteries, use 3 batteries, repeat the experiment, use 3 battery, repeat the experiment, use 4 battery. So the examiner wants to know how did you get different light intensity? You must have in the procedure. We use 2 battery, 3 battery, 4 battery. So the examiner understands, oh, this is how he changed the light intensity. He gives you 1 mark. Rate of photosynthesis. What did you measure in the experiment to get the rate of photosynthesis? Normally, the responding variable, we need to mention the device. Let's say you are measuring the temperature. You must measure the temperature using thermometer. If you are measuring the time, measure the time using stopwatch. For the responding variable, the device is important. But this experiment is unique in the sense we don't have a device. You are counting the number of bubbles by using your eyes. So the eye here, we don't count it as a device. So we just mentioned we count the number of bubbles. So we don't have a device for it. But most of the time, there need to be a device. I'm hearing some sounds outside of my room. My door is closed because I'm alone at home. Continue. Okay, so I was explaining about, counting the number of bubbles we're using eyes. So by right, there is no device. But let's say, some other experiment comes out. If there is a device, make sure you mention. For the responding variable, the device is super important. Must mention the device. By right, this experiment responding doesn't have a device. In terms of constant, make sure you use the same concentration of sodium hydrogen. Actually, in my explanation, I'm using 0.2%. Here is 2%, should be 0.2%. So make sure you mention the concentration of sodium hydrogen carbonate solution. Mention to the examiner every time you use the same concentration. Every time you use the same plant. Every time you use the same distance. Every time you use the same light bulb. Realize that many of the constant is in the diagram. Many of the constant is in the diagram. I've already highlighted for you how many? 4 constant from the diagram itself. You most probably need to explain only one of it, but I'm showing you many of the constant variables you can get from the diagram. So when you are writing the procedure, the examiner wants to know how did you get different manipulated variable? How did you get different light intensities? How do you collect the data responding variable? What do you collect in the experiment to use to calculate the rate of photosynthesis? How do you make the constant constant? What did you fix throughout the experiment? What are the ones you didn't change? All of this must mention. Since the question also mentioned precautionary measure, have at least one precautionary measure. The 4 marks is direct. MV, RV, CV, precautionary measure. That's the 4 marks. So by right, you need to have that 4 sentence. I'm going to show you the whole answer. And actually, I'm supposed to hide this. 1 minute. I forgot to hide. I need to show you one by one. There's some technical problem. I should have hit it earlier. Forgot. Okay. I erase and then I'm supposed to reattach. I forgot about it. Okay, come back here. Okay, so this is the procedure. Attach a paper clip to the top of the stem cutting of one Hydrilla sp plant. Hydrilla is a scientific name, so make sure it's in italic. I have made this whole thing in italic because English I'm using italic, BM I'm using normal wording. When you're writing this in exam, Hydrilla, make sure you underline. Hydrilla, make sure you underline. It's a scientific name. Start with H. It could be Hydrilla, it could be Elodia. Doesn't matter, both do the same thing. SP dot is short form for species. No need to underline. No need to underline. Just write the SP dot. The dot is part of the name. SP dot. That's why I have 2 dots here because this dot is the end of the sentence. This dot is part of the SP dot, short form for species. So by right now, you could have another procedure before this. obtain a high DILA stem and cut the stem. When you cut the stem, cut it across so that more surface for them to release the air bubble. Okay, this is the first step. Next step is you must place it inside a boiling tube that has 0.2% sodium hydrogen carbonate solution. Alicia earlier asked, I want to know can you show me back the constant variable? This is the constant variable. Make sure the stem cutting is facing upward. The cut stem is facing upwards. I'm using pink color for precautionary measure. Anything that is in red is the constant variable. All of this constant variable. Type of plant is constant variable. Once you already place it in the boiling tube with the cut stem facing upwards, we need to start the stopwatch. Okay, sorry, before that, you clamp it vertically to the retort stand, place the light bulb, measure the distance. So this is also red color, meaning this is constant. This is also constant. There's a lot of constant in this experiment, constant variable. Once the apparatus already set up, now you need to light up the bulb. I'm using the batteries as the manipulated variable. And I have done, in my experiment here, I have only 2 experiments, which is I'm using 2 batteries and also 4 batteries. That means you get only 2 reading. If the same question comes out, maybe they can have 4 experiments. Use 1 battery, 2 battery, 3 battery, 4 battery. We don't know. Whether there's going to be, whether you're going to use the battery for this process also, we don't know. But assuming we are using the batteries to control the light intensity, I have only 2 experiments. 2 batteries, 4 batteries. Because I feel, my personal opinion, if you have many data, then the duration is long. Maybe you can't complete the experiment. I'm using 2, maybe 3. We don't know. So maybe first time you use 2 battery, second one you use 3 battery, maybe the third one use 4 battery, or maybe just 2 battery, 4 batteries, we don't know. My experiment, I'm just using 2 and 4. So light up the bulb using 2 1.5 volt batteries. The moment you light up the battery, immediately you start the stopwatch. You need to wait around 5 minutes. You need to allow the plant to adjust to the new light intensity. They must also adjust to the new temperature. So give them around 5 minutes. After the 5 minutes, every 2 minutes, count and record the number of air bubbles released. To get the average, you need to measure 3 times. So 2 minutes, 2 minutes, 2 minutes, continuous. Every 2 minutes, the number you get, record it down. You do it for continuous 6 minutes. But before you do that 6 minutes, 5 minutes, you must allow the plant to adjust to the new light intensity. So count the number and record. See, I made this into blue because this is the responding variable. If you don't have this in your procedure, you're going to lose 1 mark. Because this is the main method how we get the responding variable. If you don't have 2 1.5 volt batteries, if you don't have that for this question, if you don't have the 2 batteries, you are not explaining to the examiner how you're controlling the light intensity. So that one must have. This is the manipulated variable. This is the responding variable. Everything else in red color is constant. The pink color is the precautionary measure. End of the day, you take note of the variables. Make sure you have it in the answer. You can do well in paper 3. Paper 3 by right, not hard. But this year, not sure. Let's see what happens. Okay. So replace the 0. When you repeat the experiment, replace the sodium hydrogen carbonate solution. Replacing is a precautionary measure because when we did the previous experiment, the carbon dioxide already used up. The plant already used the carbon dioxide from the sodium hydrogen carbonate. Sodium hydrogen carbonate provides carbon dioxide. So in the earlier experiment, maybe the carbon dioxide already finished. So we replace with a new one. So this precautionary measure. We repeat steps 4, 5, 6. Switch on the light, leave it for 5 minutes. The next 6 minutes, every 2 minutes, get the reading. And then you just record the data, use the data to calculate rate of photosynthesis. Out of the many that we have here, the ones that you must have, maybe you can take this, number 2. The ones you must have, number 4, number 5, number 6, number 7. All of this, preferably you must have, because we need at least one precautionary measure. You must have explanation. See, I need to highlight this one also. You repeat with 4 batches. So you must have explanation. If I show you the whole sequence, okay, a bit far, maybe you can see. Yeah. So explanation how you change the manipulated variable. Explanation how you get responding variable. What are the constants and what are the precautionary measures. Must have in the procedure. I think most probably for your exam is going to be guided. You might not need this, but in a better safe than sorry. We prepare for both, guided, unguided, both we prepare. So this is the procedure at the moment. Do you have any question for the procedure? If you don't have a question, you just press the raise hand button that you have in the Zoom panel. There should be a raise hand button. If you don't have question, just press the button, then I can continue. If most of you already press the raise hand button. Someone blessing me also. I'm not sure. These two things like that means what is the meaning? Someone put a devil picture also there. Oi. One got thumbs up. I wonder that there was a 2 hands pointing upwards earlier. I wonder that is for what? Female. That means female like that. Oi, Lucian. Simply making up, sorry. Okay, done. So, they don't understand. Oh, okay. No. I cannot go into that now. Okay, at the moment, we are done with the procedure. So, assuming you have conducted the experiment, the next question. So, we completed this. Okay, actually the question was here. So, we have completed the procedure. Now, we go to construct a table and record the data collected from this experiment. I'm reading here. The following title should appear on your table. Now, this was, again, we always learn from mistake. The first 2 paper 3 question, 21 and 22. 21 and 22. The table was in the question. They had the table. The student just need to fill in the data. So, out of nowhere, last year, the question wanted the student to create the table. So many students were caught unprepared because the previous 2 questions, the table was there. So they didn't take the effort to learn how to construct a table. So when the actual question came out, they asked to construct the table. Many students were like caught unprepared, and some didn't know what to write also. So here, I'm not giving you the table. I'm asking you to construct the table. That is one problem. You need to construct the table. Another thing that I'm purposely making it hard for you is, I'm asking you for the rate of photosynthesis, but I didn't inform you how to get the rate of photosynthesis. The formula for rate of photosynthesis, I didn't give. So, most probably the students are, they'll be wondering, some students, they'll be wondering, hey, how do I calculate the rate of photosynthesis? What's the formula? If you are lucky, the formula will be given in the question. They are years, they give the formula. They are years, they don't give you the formula, but they give you the clue. Most of the time, the clue will be in a form of the unit. So in here, they may be right here, bubbles per minute. So if they had the bubbles per minute here, then you know, okay, is the number of bubbles divide with the time. Normally, if they don't give you the formula, they give you the unit. They give you the unit. From the unit, you can know, you can predict what is the formula. If I say bubbles per minute, that means number of bubbles released in 2 minutes Divide with 2 minutes. Because that's the clue. But the thing is, I didn't give the clue here. My app hang 1 minute. Let me restart the app. Sir, does that mean we have to count every individual bubble? Yes. When they use the word count the bubble, what do you have in mind? If you're not counting the individual bubble, what's the other option you have? The student that are asking me this question, what's the other option you have if you're not counting individual bubbles? What other option you have? What is your the alternate plan? If you're not counting all the bubbles, then what you're counting? I'm a bit curious in the sense what you're going to calculate. Okay, haven't got the answer from the student yet. You have to count individual bubble. Must count individual bubble. You can. That is why you have to be observant. You need to count. And sometimes, if the light intensity is too high, you might have a lot of bubbles released at the same time also. If you check in YouTube, some videos you see the bubbles coming out it's like a continuous bubble coming out. If it comes out like that, very hard to count. You cannot count too many of them. So I think the light intensity, by using the different number of batteries, we can control the number of bubbles that are being released. Maybe the number of bubbles less compared to if you directly use a power source. Okay, coming back here. So as I was explaining that, how do you know the rate of photosynthesis? Formula not given, unit also not given. What I have done is I have purposely, I'm just, sometimes they do this. They will give you the clue. At least the unit they will give you. Most of the time, the unit will be here, but it's not here. So if you need to know the formula, maybe you can go to the other questions that you have. And if you go to the other question, you can check the graph question. Question F. Using the table, data in the table created for question B, plot a graph, the rate of photosynthesis. I purposely put the clue here. Instead of giving it in question B, I purposely put it in question F. Students who don't know, who are not exposed to this, most probably in question B, they'll be stuck. How do I calculate the rate of photosynthesis? We will give you the unit. The actual exam, they will give you the unit. Most of the time, it will be in question B. Sometimes it could be in question F. I'm purposely putting in F to show you. It might be in here. If it's not there, go to the graph question. From there, normally you can get the formula, at least the unit. From the unit, you know how to calculate. So once I already know the formula, I can start to construct the table. The table must start with manipulated variable. Manipulated variable, after that responding variable. It must be manipulated, after that responding. The responding, actually there is two. Even manipulated by right. There is the practical one, there is the theory one also. Practical is the number of bulb. The number of battery. Practical. Theory is light intensity. In the practical, you are changing the number of batteries. That's the practical. The theory is the light intensity. In the experiment, you are counting the number of bubbles. Number of bubbles is practical. Rate of photosynthesis is the theory. So when you are writing, when you're constructing the table, you must start with manipulated variable first. So in here, voltage of dry cell must have the unit in volt. No unit, no marks. You must start with manipulated variable. So this is the MV. This is the RV. The number of air bubbles released in 2 minutes. The unit is bubbles. This is from practical. I just put as P. This is from theory. The responding variable from theory, which is rate of photosynthesis bubbles in minute. In terms of the number of air bubbles, you need to get the data 3 times. So number 1, number 2, number 3, you get the average. I did not conduct the experiment. I did not conduct the experiment. I did not conduct the experiment. So the data that I have in here. So another thing, the voltage, there is 2. In my experiment, I only have 3 volt and 6 volt. Take note of the decimal point. Many students are going to write 3 volt, 6 volt. If you don't have 0.0, not accepted, because the battery is 1.5 volt. It's already 1 decimal point. So when you're writing 3, you must follow the decimal point, 3.0, 6.0. As I already mentioned earlier, my experiment only has 2 data. Your experiment might have 3, might have 4, I don't know. I'm assuming it's 2 data. That's why I have only 2 values. Maybe in your experiment, 3 volt, 4.5 volt, 6 volt, we don't know. I'm assuming it's 2 data. Make sure you use the same decimal point and make sure you have the unit. Actually, by right, I don't need this. I don't need this because it's already here. So I just write 3.0, 6.0 should be enough. I repeat, I didn't conduct the experiment. So I'm getting all the round numbers in here. I did not conduct the experiment. Actually, I'm just using some nice numbers so that I get a nice number in here. Just play around with numbers. You conduct the experiment and get the data. Don't blindly copy this. We don't even know whether this is the question. Don't blindly copy this. Conduct the experiment, get the data from the experiment. Even if the data you are getting feels like maybe this is not the right one. Maybe if you feel this is not the right one, don't cheat. In the sense, you are a scientist. When you conduct the experiment, in your mind, you have a certain value. Okay, when I do this, this is what I'm supposed to get. You're aiming for that. But when you conduct the experiment, you get something else. You must be truthful. That something else is what you must report. Because you wanted something else, in your mind you wanted something else. You wanted 20 bubbles. Suddenly you get only 5 bubbles, for example. You must write the 5 bubbles. You need to be a truthful. That is how scientists work. You don't cheat. What you observe is what you write. You get 5 bubbles, write 5 bubbles. Don't cheat. So we don't know what is the value. When you conduct, whatever value you get, write it. If you are using your own values, people who are marking the paper, they will know, because they also they have a certain data for reference. If you are using your own data, they will know that you are cheating. So they might not give you the marks that you're supposed to get. So we get 3 values. We get the average. In here, I'm getting a nice round number. In case in your experiment, you are getting some decimal, you can maybe leave it to, let's say, you get 20.2, for example. So make sure this one also is the same decimal point. No need to, you can use 1 decimal point or 2 decimal point. Don't go more than that. I think for this experiment, 1 decimal point should be enough. If you get both round numbers, even better. Without decimal point, even better. But if there is a decimal point, adjust accordingly, because you need to use this data to draw the graph. The more decimal point you have, the harder for you to draw the graph. So choose wisely whether you need to have 1 decimal or 2 decimal based on the graph that you're going to draw. So these values we get in means we need to do 3 times for each voltage. Yes. In the procedure earlier already mentioned what? In the procedure earlier we already mentioned. So every 2, you do it for 6 minutes. Here you do it for 6 minutes. In that 6 minutes you get 3 data. Every 2 minutes you get 1 value. So after the 5 minutes you start, you get 3 data. First 2 minutes 1 value, another 2 minutes 1 value, another 2 minutes 1 value. Yes, who's this? Ryan, yes, but later. I'll edit first before I upload into the website. At the moment, it's in private mode. You can't watch it. Later, I'll put it into public mode, but I'll put it maybe by tomorrow morning. I'll put it. At the moment, I'm doing the recording first before I edit. Okay, so coming back here, so you get the average. You get the average and follow the decimal point. In the table, the decimal point is important. Like here, you put 3 and 6, lose 1 mark. If here you have 20.2, but here you have 40.40 only, without the 0, consider not following the decimal point. Must follow the decimal point. What are the values in here? I don't know. Conduct the experiment. Is it going to be 2 data or is it going to be 3 or 4? I don't know. Adjust accordingly. So the rate of photosynthesis in here is easy. 10 and 20. 20 divide with 2 minutes. 40 divide with 2 minutes. Bubbles per minute is basically the value. So this is the table. The thing I want to highlight here is manipulated variable, after that responding variable, the practical version, after that the theory version, in that order. Don't forget the units. Without the units, the table is incomplete. Make sure you have the same decimal point. These are the things you need to take note when you're constructing the table. If you're lucky, the table already there. You just need to fill in the data. Sometimes, like last year, they ask you to construct the table. So in case they ask you to construct, this is how it's supposed to be. We have given the procedure. We have conducted the experiment. We have completed the table. After this is all standard questions. What is the inference? What is the observation? What is the inference? What is the hypothesis? Make some prediction. Give some definitions. Standard questions. So next one. Based on table 1, state one observation. Satu sahaja. Observation means you write back what you see. What you observe, you write back. No need to make any, no need to do any explanation. Just write what you see. My experiment has only 2 data. Because I only have 2 data, 3.0, 6.0, I can choose either one. Because my experiment only has 2 data. So got too excited, I scribble here and there. Now I have to remove each one of it one by one. Just remove all of this. Okay. My experiment has only 2 data. So I need one observation. I can use either one. I can use 3.0. I can use 6.0. But maybe let's say your experiment, maybe in your experiment is 1.5 volt, 3.0 volt, maybe 4.5 and then 6.0. Let's say it's like this. Meaning you have 4 data. If you have 4 data, which one you use for your observation? Use the responding variable. The responding variable that has the lowest value and the highest value. And it's not always the first and the last. It's not always, that means encourage you to use the lowest or the highest responding variable. The lowest and the highest. Either one of it, either the lowest. Normally the better one is the highest. The highest responding value, responding variable, use that as the observation. That will be the best observation. If you have more than 2 data, if you have only 2 data, any one of them is fine. More than 2, preferably look for the responding variable that has the highest value, and it doesn't have to be the last one. For example, if you have a temperature experiment, enzyme, 0 degrees Celsius, maybe 15, maybe 37, maybe 45. The graph looks something like this. The last experiment is not the highest value. This is the highest value. So a good observation should use this as the observation. Why? Because it has the highest MV. Always look at the MV. No, sorry, not MV. RV. Too excited. Okay. RV, this is MV. It has the highest RV. So normally it is the last one. Sometimes it's not the last one. So in your experiment, most probably we don't know 2 data or more. If it's more, use the highest RV. If it's only 2 data, you are free to use any one of it. That's why here I'm going to use one of it. And what you must have in your observation standard, you must have MV, you must have RV. That's all. Make sure and you must have the data. These 3 must be in the answer. And where you get that? Make sure in your answer you have voltage of dry cell. Make sure in the answer you have the number of air bubbles. This wording that you have here, make sure you have it in the answer. The voltage of dry cell and also the number of air bubbles, you must have it in the answer. These 2 you must have in the answer. So my observation, my first observation is this. When the voltage of dry cell is 3.0 volt, average number of air bubbles released in 2 minutes by hydrilla SP is 20 bubbles. What I've given in purple, manipulated variable. What I've given in blue, responding variable. What I've given in red is constant variable. What I've given in purple, in pink is the data. I have everything the examiner needs. I get the 1 mark. MV, RV data. If you can have the CV, also good. In here, 2 minutes is CV, constant. Hydrilla is constant. Just make sure you have MV, RV data in the answer. You can use 3.0. In my experiment, I'm free to use either one. I'm using 3.0. I can also use 6.0. Both of them can. Give me 1 minute. Okay, this is the observation. Standard after observation will be inference. Inference is basically, you just told the examiner you counted 20 bubbles if the dry cell is 3 volt. The examiner wants to know what is the explanation. Oh, sorry. Okay, I repeat. Standard after the observation is inference. What is inference? You just told the examiner you get 20 bubbles when you use 3 volt. So the examiner wants to know the scientific explanation. What is that bubble? And what is that 3 volt battery? What does it control? So you are explaining it scientifically. This is where you need to use the theory explanation. The voltage represents the light intensity. The number of bubbles represents the rate of photosynthesis. So you need to give the scientific explanation. Because I have only 2 values. Because I only have 2 values in here. I'm just going to use the word high and low. I'm just going to use the word high and low because I have only sorry. I was showing here earlier. I repeat the whole explanation. Okay. This is observation. Now, after the observation, standard question is inference. Inference is basically, what is your scientific explanation for the observation you gave? You told 20 bubbles. The voltage is 3.0. What is your scientific explanation for it? So this is where we bring in the theory part of it. The light intensity and the rate of photosynthesis. You can, by right, go back to this table that you created earlier. Since I have only 2 data, I'm just going to use the word high and low. This is low light intensity. This is high light intensity, low and high. If you have more than two, let's say, like earlier, you had more than two, maybe you can use the highest and the lowest. If I have only two data, I cannot use the word highest, lowest. I use high and low. If I have three or more, I can use the word highest or the lowest. At the moment, I'm using high and low. I repeat, because I have only two data. If I have more than two, I can use the word highest and lowest. At the moment, I cannot because I have only two data. So what is my inference? Light intensity is low. High Dila SP rate of photosynthesis is low. See that this is the theory. This is the practical. Voltage of dry cell is what you did in the experiment. The practical part of it. The theory part of it is the light intensity. The number of air bubbles released is the theory, practical part of it. The theory is the rate of photosynthesis. The rate is low is the data. So I'm just adding in another sentence in here, which is less oxygen is produced. I'm explaining to the examiner what is that air bubble. Less oxygen gas is produced in light dependent. We are bringing in the theory. Photosynthesis is divided into light dependent, light independent reaction. Read back chapter 2 from 5. The oxygen is produced in the light dependent reaction. Light independent, the second part uses carbon dioxide. The first part is where oxygen is released. So less oxygen gas is produced in the light dependent reaction of photosynthesis. We are giving the theory explanation of the practical. Many times you can also refer to the question right at the beginning. This is the theory, light intensity and rate of photosynthesis. We are just using back this in our explanation for inference. I repeat, I have two data, I'm using low and high. The moment you have the third data, you can start to use the word highest, lowest. Light intensity is highest. Rate of photosynthesis is highest. If you have three or more data, I'm using only two data. After the inference, the next question. Identify the variable and how did you control the variable? Because it's guided, this is the question. If we don't know guided, unguided, most probably guided. If it's guided, this is the one that you need to know. What is the variable and how did you control the variable? MV, manipulated variable. Voltage of dry cell. That is the manipulated variable. You need to inform the examiner what did you do in the experiment to get to control the manipulated variable. So you must have the word use. You must have the word use. Without the word use, no marks. Use different number of 1.5 volt dry cell batteries. And preferably inform what did you use, which are 2 batteries and 4 batteries. Use different length. Use different length between the plant and the light bulb, because normal experiment you change the distance of the light bulb, 20 centimeter, 30 centimeter. If that is the experiment, then your manipulated variable is the distance. Use different intensity. No, the light intensity is the theory. This is the practical. This is the practical. You must write what you did in the practical. The light intensity is what you calculate later. This is in the experiment. They already mentioned in the experiment. So you can't be writing the theory in here. You must write the practical in here. Theory cannot. So you cannot write here a light intensity. You must write voltage of dry cell. The student is asking the question. Cannot write theory because this is what you did in the experiment. So you must use the word voltage. Generally, what you had in the table here, this one, and this one. This is what they want to see in the answer. Because this is the theory. This is the last one here is the theory. We are interested with what you did in the experiment. So what you did was you changed, in my experiment, I'm changing the number of batteries. Maybe changing the distance, we don't know. Check the experiment, what you're supposed to do. But the thing is, you must use the word use. This is the variable. This is how you control the variable. Responding variable. Corresponding variable is number of bubbles released in 2 minutes. That is the responding variable. How did you get the responding variable? Count and record. The number of air bubbles released in 2 minutes. Count and record must have in the answer. You don't have the word count and record, consider the answer is incomplete. Count and record number of air bubbles released in 2 minutes. In here must have the word use. In here must have the word count and record. Responding variable, there's another answer. There's another answer, which is this. You can also use rate of photosynthesis as the answer. Can. But if you are using the rate of photosynthesis, you need to use this as the answer. Calculate. You must have the word calculate using this formula. Calculate using this formula. If you want, you can use this as the responding variable, or you can use this as the responding variable. The student that asked me the question earlier, so what if I use light intensity? If you are using light intensity, here, can. But why I say earlier cannot? What is your calculation? If I write here light intensity, let me bring this side. Okay, I write here light intensity. If you want to use light intensity, so this is light intensity, yes. Can. It's a correct answer. But do you have the explanation in terms of the formula? Other. That is why we are not using the light intensity. If you have a formula for light intensity, you can use. If not, you can't explain. You can give the examiner light intensity, but you can't explain how you get the light intensity. So this is not the right choice because we don't have the information. If the actual question in your exam has a formula for light intensity, there is a formula. Check the question. If they give you the formula, by all means you can use. Just use this word. Calculate light intensity by using this formula and then give the formula. But compared to that, you have a simpler answer. Why complicate our life? This should be enough. That is also correct. But you must have the data. I know the formula for light rate of photosynthesis. I don't know the formula for light intensity. So that's why I'm not using it. So the student that asked the question earlier, is that okay? I'm not mentioning your name. Is that okay? The student that is asking me, sir, can I write both? Why you want to complicate your life? They only want one answer. The best is always the practical. The best answer is always the practical answer. As much as possible, try not to, because this is paper 3, it's about practical. So as much as possible, give answers related to the practical. Ini pun boleh. Ini boleh. Betul. But I don't encourage you to use this because as you can see, it's complicated. Susah untuk, you have to write the formula. Just use the practical part of it. Now make your life easy. Make the examiner's life also easy. Encourage you to use practical. Don't write both. Choose the practical as much as possible because practical, surely you know the answer. Theory, like light intensity, kita tak tahu. So don't complicate your life. Just use. I'm showing you it's possible to write this. But I'm not encouraging you to write. I'm asking you to write this. That is, if this question comes out, we don't know. Next question. After you get the variable, constant variable, you must use the word fix. Now, constant ada banyak jawapan. It's a lot of answers for constant. I told you 8 o'clock, right? Sorry, I misjudged the timing. A bit more, a bit. I thought can finish in 1 hour because I have another class at 8:30. Oh, okay. Type of plant. That's one of the easiest type of plant. Any constant, make sure you have the word fix. Fixed type of plant as Hydrilla sp. I think that's the only one I have here. You have a lot of other answers this side. There's a lot of other answers. All of these are constant. Distance between light bulb with Hydrilla. Fix it at 20 centimeter. Concentration of sodium hydrogen carbonate. Fix it at 0.02%. Power of light bulb. Fix it at 60 watt. Number of Hydrilla. Fix it at 1. All of these can be used as constant. You decide which one you want to use. But in the answer must have the word fix. For constant must have the word fix. For responding must have the word count and record. For manipulated must have the word use. The one I put in green, that's the keyword you must have in the answer. Next question is hypothesis. Hypothesis normally is just combination of higher or lower, higher or lower. The normal answer, the higher the light intensity, the higher the rate of photosynthesis. Higher, higher. Maybe higher, lower. Maybe lower, higher. It depends. Most of the time is higher, higher. Or maybe higher, lower. The higher the humidity, the higher the humidity, the lower the rate of transpiration. Sometimes opposite, sometimes follow. Most of the time follow. The higher the light intensity, the higher the rate of photosynthesis. But the word higher, 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 lower, you can only use if you have more than 3 data. More or equal to 3 data. If you have 3 or more, you can use higher, 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 lower. If you have only 2 data, my problem is I have only 2 data. I don't know. I'm expecting this question, so I'm using only 2 data. 2 data, I cannot use the word higher, 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 lower. If let's say your experiment have 3 or 4, you can use this. I'm using only 2. Because only 2, I must use the word compare. I must use the word compare. Hypothesis also, you can use the theory and the practical. But hypothesis is always easier if you use the theory. What was the theory? Light intensity, rate of photosynthesis. For hypothesis, encourage you to use the theory. So at high light intensity, rate of photosynthesis is high compared to low light intensity. Compare. Why I'm using compare? Because I have only 2 data. If I have more than 2 data, I can use this answer. The higher the light intensity, the higher the rate of photosynthesis. I'm showing you the answer, but the rule is minimum 3 data. Since mine has only 2 data, I'm going to use this as my hypothesis. You can also use the practical. You can also use the practical in here. The higher the number of dry cells, the higher the number of dry cells, the higher the number of air bubbles released in 2 minutes. Boleh. You can use theory, you can use practical. But you can see, the moment you use practical, it's a very long sentence. The higher the number of batteries, dry cells, the higher the number of air bubbles released in 2 minutes. Very long. For hypothesis, always use the theory to make our life easy. The higher the light intensity, the higher the rate of photosynthesis. Easy explanation. So this will be for hypothesis. Next question. Okay, this is, I'm just doing a bit of commercial break. By right, you should know, since you are in checkup, also a formula. My normal classes I already convert into seminar. Some of you are still not sure what I mean by seminar. My seminar has 4 exam sets. So the 2 classes, not the 2 months of December and January, I'll be discussing 32 structure question, 12 essay questions that I feel important for the exam. So all of these questions will be recorded. The recording will be in the website. You can log in anytime to watch how to answer the questions. Total is around 30 hours. Based on what I did last year, 90% of the actual SPM exam question, 90% of the question we already discussed in class. So by right, if you attend the seminar in code 2 months of seminar, you should have you can confidently go for the exam. We have covered almost everything, and we have done this number of questions. By the time you go for the exam, you can confidently go for the exam. If you're interested, you can just ask the center. In case you don't know, you can just ask the center how to join this class. Okay, next one, using how to join this class. Okay, next one, draw the graph. My graph only have 2 data. Because my graph only have 2 data, I have drawn a histogram. If you have 3 data, then maybe let's say the 3 data is like this, then you draw like this. It's based on the number of experiment. Mine is 2 data, so I'm just using a histogram. In the graph, what they want to see. Do you have the heading, dry cell voltage? Do you have the unit? Is your scale correct? The distance must be same. For every 2 is 1. So the scale must be same. Do you have the origin? Many students forget to write the 0. If your heading correct, one minute, Sharon, let me finish this first. Okay, if your heading is correct, your scale is correct, you have 0. You get 1 mark for the x-axis. The same rules applies for the y-axis. Do you have the rate of photosynthesis and the unit? Do you have the scale, standard scale? Try to make the largest graph possible. If you see this is the graph paper, I have almost as large as the graph paper. You get 1 mark for the x-axis, you get 1 mark for the y-axis, 1 more mark is for the graph itself. Most students, they are preoccupied with this. They are interested with this one. They spend less time on the x, they spend less time on the y. End up, they lose the x and y marks. Secure your x, secure your y. Make sure it's complete. After that, only start to draw the graph. Spend some time. At the same time, don't waste too much time on the graph. The thing is, make sure your x is correct. The heading, the unit, the 0. Make sure your y, the scale, the heading all correct. After that, only start to draw the graph. Because mine is only 2 data, I have drawn a histogram. 2 data cannot draw a line graph, so must draw a histogram. Let's say you have a lot of data. If you have 3-4 data, standard, make sure you follow x and y. If you have, let's say, graph, these are the 3 points. So you can make it a smooth curve, example, like this. Or if you, sometimes you might have to do the best fit. For example, maybe you have 1, 2, 3, 4. So maybe you have to draw a line like this. Best fit, there is 1 above, there is 1 below. Maybe something like this, 1 above, 1 below. But if you draw like this, make sure it ends here at the same line. It begins at the here. For bio, you cannot simply extend. Some students like to extend the line to 0. Maybe for other subject, yes. Maybe for other subject, yes. For bio, you extend the line, not accepted. You only draw what you know. Either you draw a best fit or you draw a smooth curve. Most probably, if you have more than 2 data for this experiment, most probably the graph will be something like this. By right, and maybe you have 1 data here, 1 data here, 1 data here. Maybe you have 4. It should be increasing and then should become constant for light intensity. But it all depends on how many data you have. Don't extend. Don't simply extend the line to 0. For bio, cannot extend. Stop. Start at the first point. Finish at the last point. Don't extend both sides. Make sure you label. Make sure your scale is correct. Don't forget the zeros at both ends. Here also same thing. Should be enough to get you the 3 marks. Title for us, no need. Since you're asking, better step, then sorry, write the title. Write the title. Since you're asking, write the title. As far as I know, no need the title. But since you're asking, we call that, I normally call that as the sixth sense. If something is telling you, something is asking you to ask me, sir, do I need to write the title? That's your sixth sense asking you to do that. So write it. They're not going to minus mark. Write it. Maybe here cannot because my label is here. You can always write the heading. So heading, you can follow what you learn in physics. You can write the heading. But don't extend. My graph only have 2 histogram because I have 2 data. You can check your graph, draw accordingly. Okay, so once you already draw the graph, next question. Based on the graph, explain the relationship between rate of photosynthesis with light intensity. So, rate of photosynthesis with light intensity. The answer that I have here, the higher the dry cell voltage, the higher, I'm explaining the graph. The higher the dry cell voltage, the higher the light intensity. Take note, this is from the graph. This is the theory. From there, if I go down, rate of light dependent reaction of photosynthesis is higher. More oxygen gas is produced and released. More glucose are produced. Rate of photosynthesis is higher. Okay. We relate what we have in the graph. In the graph is the number of dry cell voltage. As the dry cell voltage increase, light intensity increase. When light intensity increase, how come the rate of photosynthesis increase? You need to explain. When the light intensity increase, why the rate of photosynthesis increase? You need to have the theory explanation. That is what I'm doing here. When the dry cell voltage increase, light intensity increase, yes. The rate of light dependent reaction of photosynthesis is higher. When the light intensity is higher, light dependent reaction rate is higher. They produce more oxygen. The more oxygen explains why we get more bubble. So more glucose is produced, the rate of photosynthesis is higher. So the explanation that you're giving is something that you don't have in the graph. When the voltage increase, what increase? Light intensity increase. When light intensity increase, more oxygen. When more oxygen is released, that means photosynthesis rate is higher. Normally you explain by using what is not in the graph. In the graph, I have dry cell voltage. What I don't have, light intensity. I put it in the answer. In the graph, I have rate of photosynthesis. What I don't have, the number of bubbles, is oxygen bubble. So more oxygen bubble. What's not in the graph, use it as the explanation. That will be for this. One of the last questions we have is prediction. Okay, now prediction part. The prediction part is basically throughout the experiment, constant was constant. You didn't change the constant. Prediction is the question where we change the constant. Most of the time we change the constant. So there's a lot of things I can do here. I can change the concentration of sodium hydrogen carbonate solution. I can change the distance. I can change the power of light bulb. There's a lot of things I can do. In here, what I've decided is I am changing the number of hydrilla. So instead of one hydrilla, now I have two hydrilla. I'm asking you to predict the result. When you predict the result, you must use data from the table. You must use data from the table, meaning which one you must use. Either this or this in your answer, or this and this in your answer. This one obviously you must have lah. You can choose either one. As I mentioned earlier, if you have more than two data, preferably choose the lowest or the highest MV. More than two data, choose the lowest or the highest MV. RV. Lowest and highest RV. If you have more than two data. In here, I have two data. I can use either one. If it's one plant, it's giving me 20 bubbles. Two plants should be more than 20 bubbles. I need this value 20 in the answer. If one plant giving me 40, two plants must give me more than 40. The 40 must be in either one. 20 or 40 must be in the answer. The data must be there. Most of the students, what they do, rate of photosynthesis become higher. They give a general statement. Rate of photosynthesis become higher. They won't relate to the experiment. In here, you need the data. Make sure you have the data. When the voltage of dry cell is 6 volts, average number of bubbles released in 2 minutes by 2 hydrilla SP plant is higher than 40 bubbles. The 40 bubbles is from the data. This is from the data. You must go back to the table when you're making the prediction. What else I have in the table? I have the rate of photosynthesis. If I want, I can give that as the answer. Rate of photosynthesis become higher than 20 bubbles per minute. No need to explain. You are predicting the results. When you predict, you must use the data. So preferably write both. If possible, write both. The observation and also the calculation. Both write in the answer. If you have more than 20 bubbles, if you have more than 40 bubbles, more than 20 bubbles per minute, more than 40 bubbles should give you the marks. Why? This is data from experiment. Prediction question. Last question is operational definition. Oh, sorry. Before that, you need to explain your prediction. When I'm using 2 hydrilla, that means I have, maybe you can write here, there is more leaf, there is more chloroplast. I just wrote here more chloroplast. You can even use the word chlorophyll for this. More chlorophyll. Okay. You can write there is more leaves, there is more chlorophyll, more light is absorbed. More leaf, more chlorophyll, more light is absorbed. The rate of light dependent reaction increases. More oxygen gas is produced, more air bubbles are released, rate of photosynthesis higher. This is the theory. I'm changing the number of plants. They might change, maybe they use higher concentration of sodium hydrogen carbonate. That means more carbon dioxide. Maybe they use a higher powerful light bulb, or maybe a smaller power light bulb. The light intensity higher, lower. Maybe you go back to this, the constant. We had all the constant earlier. One of these they are going to change. Maybe the distance they change, maybe the concentration they change, maybe the power, maybe the number, or even maybe the type. I don't think they change the type. It should be one of these. I'm just changing the number of hydrilla plant. Which one they are going to change, we don't know if this experiment comes out. So that was the explanation. Last question. Last question is operational definition. Many students will come and ask, so how do you write operational definition? Operational definition means based on this experiment, what is photosynthesis? So you don't give the theory answer. Process that produce organic substance from inorganic substances by using light energy absorbed by chlorophyll. Itu theory. Many students will write the theory. You are supposed to write, in this experiment, how do you know photosynthesis occurred? I know the plant is doing photosynthesis because I see the plant releasing bubbles. That is the main point. In this experiment, photosynthesis is a process that releases air bubble from the hydrilla plant. Ini pun mesti ada MV, RV, CV. This is the last question in the experiment. The last question, take it as a summary. Put everything inside. MV, RV, CV. Semua kena ada. So the answer, the main point is RV. Number of bubbles released. Remove the number. Bubbles released from the plant. That is photosynthesis. See the answer I have here. Already 8:35. Okay. That means the class is what? 2 hours. 1.5. Okay. Photosynthesis is a process that releases gas bubble from one hydrilla SP plant placed in 0.2 sodium hydrogen carbonate solution that is exposed to 60-watt light bulb set at a distance of 20 centimeter from the plant. Can anyone tell me what I'm... So when we draw a curve, is it necessary for the graph to intersect all the points, or we can just draw it smoother? The general rule is you must, if it's a curve, it must be a smooth curve, follow all the points. You're only going to have 3 or 4 points, so shouldn't be a problem for you to get a smooth curve. Bio, normally the graph won't be that hard. Most of the time it's an easy graph. So you won't have a difficulty of getting a line that passes through all the line. Most of the time they'll pass through all the points. Bio graph normally not hard. Okay, in here, can anyone tell me what I'm trying to do in this answer here? What I made sure is in the answer. What I made sure is in the answer to get me the marks. Yes, correct. When Binja. Anyone else? What is the what's the word to use? What I have to make sure I have in the answer to get the marks? Yes, correct. Isa, I think so. Dushan. MV, RV, CV must have in the answer. This is the last question. Put everything inside. Photosynthesis is a process. See, I put the process green color. Actually, we can make this larger. Okay. Process that releases gas bubbles from one hydrilla SP plant placed in 0.2 sodium hydrogen carbonate solution that is exposed to 60-watt light bulb set at a distance of 20 centimeter from the plant. Okay, now I'm just putting this separately. I'm just putting this photosynthesis is affected by light intensity because I feel this sentence already long. So I'm just putting this separately. Now some teachers might claim, this one must be one sentence. If they insist on making this one sentence, then all you have to do is just put a comma here and then just change this to this process. No, some teachers will say, oh, must be in one sentence. So this process is affected by light intensity. Doesn't matter. I prefer to put it as a separate sentence. But if your teacher insists on having this in one sentence, then maybe what you can do, you can just write comma, this process affected by light intensity. The last question, pink color is the data. Release of gas bubble is responding variable. Everything that is rate is constant variable. Light intensity is manipulated variable. You must have the word process. So this will be the last question. It's like a summary for the whole experiment. Don't give the answer based on theory. In this experiment, what is photosynthesis? The main answer is responding variable. The main answer is responding variable. Make sure you have that. Add in all the constant. You should have a good answer. So that should be the last question. So take note, the question I've given here, I have combined both unguided and guided, all the possible questions. I've combined it into one. By right, in the actual exam, you will have less questions than this because I'm giving you both answers, guided, unguided, all the possible questions I'm giving you. Like, for example, prediction and operation definition, only one of it will come out for your exam. So it's either one of it. I'm doing a revision. I'm giving you all the possible questions. End of the day, it's about variable. It's about variable. Make sure every single answer you have MV, RV, CV. Accordingly, you must have, and some of the questions you must have the data. The good news about paper 3, all the answers is already in the, all the answers, all the clues you need is already in the question. So what's in your hands? Conducting the experiment, getting the data. Do it properly, get the data. From the data, answer all of these questions, get full marks. No excuse not to get full marks. I don't know whether this is the question that's going to come out. We really don't know what's the question. I'm predicting it like this. How it's going to be, I really don't know. Hope the same thing comes out. You have 1 day break before your exam, so use the time. Don't depend only on this experiment. Also read up on the other 2 experiments that you have in your textbook, in case they suddenly modify the question. Okay, so hopefully wait 1 minute. So no need to start the graph of origin. No, unless you have a data 0 0. If you have data 0 0, you start from origin. If you don't have data, don't extend the graph. Beginning, ending, use the data that you have. This recording is recorded, is in YouTube, but at the moment it's private. I'll edit the video, or maybe I think I don't edit. I just later I'll just put it in the website. I'll put the link. Then if you want, you can watch it again. Okay. So thanks for coming. All the best in your exam. I need to start my next class. All the best. If you have any question, just message me.